Yes, welcome. Good morning, everyone. So today we are going to discuss about antigen, antibody, and antigen-antibody interactions. Okay. All right. So before we proceed with this antigen and antibody, let's talk about immunity. All right. So immunity is the ability, ability of the body to recognize and destroy foreign or dangerous macromolecules. Okay. Now, examples of macromolecules are like toxins. Okay, these toxins may be produced by the pathogens like bacteria because they secrete waste, and their waste is toxins, right? Considered as toxins, and these toxins apparently they are example of macromolecules. Okay, so there are two main types of immunity, which is we have the innate immunity, okay, which will give out innate immune response, okay, and then we have the adaptive immunity, which will give out the adaptive immune response, right? Now, innate means non-specific, while adaptive means specific. So, innate immune response, also known as non-specific immune response, while adaptive immune response, also known as specific immune response. Okay. Now, this innate immunity, all right, apparently all animals has innate immunity, right, or exhibit innate immunity, while for adaptive immunity, right, only for vertebrates. Okay, so for non-vertebrates, they don't have adaptive immunity. Okay, so to conclude that for vertebrates, right, vertebrate has innate immunity and adaptive immunity. Non-vertebrates, they only have the innate immunity. Okay, now this adaptive immunity, right, okay, also known as acquired immunity. Okay, right now, innate immunity. So innate immunity, okay, is a defense, right, or defenses that provide immediate protection. Immediate protection, so meaning to say the response, okay, of the body against the invading pathogens are rapid response. Okay, so defenses that provide rapid response or immediate protection against pathogens. Okay, examples of pathogens are bacteria, fungi, and viruses because they cause sickness, so they are known as pathogens. Okay, other than pathogens are parasites, okay, and then toxins, okay, and drugs, as well as cancer cells. Okay, so innate immunity provide our body against these incoming dangers, right? Pathogens, parasites, toxins, drugs, and cancer cells. Okay, phagocytosis. Okay, phagocytosis, you know already, phagocytosis are exhibited by macrophage, right? Macrophage. So phagocytosis and inflammation will destroy bacteria, okay, when they manage to penetrate the outer defenses. And inflammation may trigger adaptive immunity. So meaning to say, okay, innate immune response, okay, may trigger the adaptive immune response, okay, via inflammation. Okay. Now, from innate immunity, we have innate immune response. Okay, innate immune response, like what I've mentioned earlier, or non-specific immune response. Okay, and this innate immune response, okay, innate immune response defined as mechanism such as barrier defenses. You can see from here, such as barrier defenses, okay, and internal defenses. barrier defenses and internal defenses that provide immediate and general protection against pathogen. Okay, immediate and general protection against pathogen. Okay, class, normally in the exam they might ask you to compare between innate immunity and adaptive immunity, right? So one of the key point is for innate immune response, it gives out rapid response, okay, or immediate protection against pathogen. Right now, example of these barrier defenses, right, are like skin, foul skin, which gives protection against huge size of pathogens, for example, okay, or from worms, for example, okay, and then secretions, which is from our saliva that has lysosomes. Those lysosomes inside our saliva, it kills some pathogens, okay, and then mucous membranes, which is secretion of this mucus from some of our epithelial cells. This mucus, right, it provides protection and it kills some of these pathogens. And these mucous membranes, right, can be found on the epithelial lining of our digestive and respiratory system. Okay, so that's why our respiratory lining or respiratory tract and digestive tract, they are very how do I say slimy in a sense. The cells there, the epithelial cells there, secretes mucus. Okay, now the internal defenses, right, the internal defenses examples are the phagocytic cells. Okay, when it comes to phagocytic cells, the first thing that rings into our mind is the phagocytes, right? You can see the phagocytes, right? They are macrophage that engulf pathogens, right? Now this macrophage, right, generally they are known as this macrophage, generally they are known as macrophage, right? When this macrophage, they settle down at certain organs of inside our body, these macrophages and known as specific name for that particular organ. For example, okay, macrophage that settle down in our liver, all right, inside the liver. This macrophage, these macrophages are known as the cover cells. Okay. Now you can see the other example of cells here that gives internal defenses in the innate immune responses. Right, we have the NK cells. NK stands for natural killer cells. These are large granular lymphocytes, and these NK cells, they are very active against tumor cells and cells that are infected with certain virus. So meaning to say these NK cells, all right, it will directly kills, all right, and destroy cells, our body cells that are already infected with certain viruses, right, as well as the tumor cells inside our body. Okay, and then we have these dendritic cells, right? Dendritic cells that is located, okay, inside all the tissues of our body that come into contact with the environment, such as the skin. So inside our epithelial skins, right, epithelial scale cells of our skin, we have these dendritic cells. Okay. Now, inflammatory response, like what I mentioned earlier. I mentioned that this innate immune response may trigger adaptive immune response via inflammation. All right. So this inflammation, right, is defined as the body's key to responses. Sorry, body's key responses to infection or injury. That's why whenever we have injury, right, or infection, we have inflammation. Usually we have inflammation, right? But there are some certain condition we don't have inflammation. So I mean to say maybe the infection or the injury is not severe, right? To the extent that it does not trigger inflammation. So this inflammation includes vasodilation of the blood vessel, okay, increased capillary permeability, and increased phagocytosis. So whenever we have inflammation, phagocytosis activity increases in that particular area. Right? And then the clinical characteristic of inflammation is fever, swelling, and redness, as well as pain at the infection site. Now, adaptive immunity, right, which only exhibited by vertebrates. Okay, now this adaptive immunity is defined as specific defenses, right? Specific defenses that target distinct macromolecules or antigens. If we compare with the innate immune response, the innate immune response targets are more general, right? But for adaptive immune response, it gives specific defenses, okay, that target distinct or recognized macromolecules or antigens. Now, from adaptive immunity, we have adaptive immune response, okay, or adaptive immunity causes adaptive immune response. Now, adaptive immune response is a defense mechanism that targets specific macromolecules associated with pathogen. Okay, so this adaptive immunity includes cell-mediated immunity and antibody-mediated immunity or humoral response, right? Now, this adaptive immune response are activated, okay, after the innate immune response via inflammation. And this adaptive immune response develops more slowly or give up slower response and enhanced by previous exposure to the infecting pathogen. So for our body to trigger adaptive immune response, innate immune response must takes in place first, right? Okay, next, antigen. Antigen, okay, they are foreign molecules that are recognized as non-self that stimulates an immune response. Okay, non-self means it doesn't come from our body. All our body cells, okay, our body recognize as self, okay, as self cell, okay, cells that are produced by our own body. So anything or any cells or any particulate that doesn't comes from our own body, our body will recognize them as non-self, and this will stimulate immune response, right? So the foreign molecules, right, which is defined by the antigen, okay, they can be protein, carbohydrates, or glycoprotein. Okay, if you recall, glycoprotein is the combination of carbohydrates with protein. Now, this antigen can be found on the surface of pathogens, all right, on the surface of this bacteria, virus, or fungi. That's where this antigen can be found. Okay, or on the surface or in the toxins itself secreted by the pathogens. Okay, and this antigen also can be found on the surface of transplanted cells. Okay, transplanted cells from the organ transplant. Now, antigen is very specific. in stimulating immune response because of the specific amino acid sequence in the antigen. Right? Recall antigen is a foreign molecule, okay, that has protein, right, which recognized as non-self that stimulates the immune response. So since this antigen has foreign molecule that made up of protein, right, and you know protein, okay, comes from amino acid as its monomer. And when it comes to the amino acids as the monomer, these amino acids must be in specific sequence. That's why antigen is very specific in stimulating immune response. Okay, again because of the specific amino acid sequence in the antigen. Now, this is the antigen. Right, you can see the antigen here, or this is the antigen which is on the surface of the pathogen. Okay, you can see the antigen here. Now, this antigen, right, it has epitope, okay, or antigenic determinant. Now, epitope is the specific part of the antigen that is recognized by an antibody or T cell receptor. Okay, for T cell receptor, we go on that part later on. Now, an antigen, okay, an antigen might have many epitopes on its surface. So several kinds of antibodies may bind to a single antigen. As you can see in this diagram here, right, this particular pathogen, right, it has antigens, right? And as what I mentioned earlier, one antigen might have several epitopes. For this given diagram, this particular antigen has two different types of epitopes. Okay, and in this diagram here also you can see that this antigen has many types of epitopes or antigenic determinant. Okay, now antibodies. Okay, antibodies are immunoglobulins. Okay, double M here, immunoglobulins. Okay, or capital I, small letter G. Okay, immunoglobulins, immunoglobulins that recognize and binds to specific antigen. That is antibody, right? They are globular glycoprotein that recognize and bind to the specific antigen. So this antibody, as you can see in this diagram here, consists of 4 polypeptides, right? One, two heavy chains, and these heavy chains also known as H chains, right? Consists of 2 H chains or 2 heavy chains and two short chains or 2 light chains, okay, or 2 L chains. Now, each of these chain, right, they are linked by disulfide bridge. Okay, you can see here this light chain with this heavy chain, they are linked with this disulfide bridge. The same goes on the other side. This light chain and this heavy chain, they are linked by disulfide bridge. The same goes for the between the heavy chains. Okay, this heavy chain with this heavy chain, they are linked with this disulfide bridge. And then each chain, right, each chain, the heavy and the light chain, they have constant region and variable region, right? You can see here for this heavy chain, this is the variable region and this is constant region. For the light chain, this is the constant region and this is the variable region. Okay, now you may be wondering what is this variable and constant region, right? So this variable region, right, ujung ni nampak kan, ujung kiri kanan ni, right, they compose of many amino acids, right? And because of these many amino acids, this give the antibody its specificity for binding antigen. So means to say at this variable region it has antigen binding sites. Okay, specific for this antibody to bind with specific antigen binding site. Now, this constant region here, okay, the keyword here is constant. All right. So this constant region determines the mechanism used to destroy the antigen. And from here, okay, we can divide antibodies into 5 major classes. Okay, they are the IgG, okay, capital I, small g, capital G, IgM, IgD, IgA, and IgE. Okay, so these antibodies are divided into these 5 groups based on their constant region structure, right, as well as their immune function. Okay, these antibodies, they are released by a mature B cell, okay, that acts against a particular antigen. Right, this mature B cell, also known as plasma cell. Combination of antigen and antibody activates several defensive responses. Okay, so when this antigen combined with this antibody, sorry, I should have mentioned, when this antibody combine with the antigen, it will form antigen antibody complex, and this antigen antibody complex activates several defensive responses, right, such as, okay, stimulate phagocytosis, where phagocytic cells ingest the pathogen, and then this antigen antibody complex also activates, right, another defensive responses, okay, that does not destroy an antigen directly but inactivate the pathogen or its toxin. Can you get it? So meaning to say, when this antibody bind with the antigen, right, it will form antigen antibody complex, right? Now, with the presence of this antigen antibody complex inside our body, right, okay, this complex will either stimulate the macrophage, okay, to ingest the pathogen, or, right, or the the pathogen will be deactivated. Okay, the pathogen is being deactivated, so meaning to say inside our body that particular pathogen is still present, except that it is not active. Dia duduk diam-diam saja. Why the pathogen being inactivated or deactivated? Because the pathogens antigen is already bind with the antibody. So the binding of this antibody with the antigen that forms the antigen antibody complex, it causes that particular pathogen cannot be active. Now this IgG and IgM, right, they works, right, mainly through this complement system. So we need to say usually this IgG and IgM will bind with the antigen to form this antigen antibody complex, right, that will stimulate one of these two. Usually there are 5, okay, so one of these two two given examples here, mechanism or responses. Okay, so now let's take a look at the classes of the antibody: IgG, IgM, IgD, Ig, and IgE. Right. So general structure of this IgG is a monomer. This is the monomer. Right. The general structure that you have seen earlier. Okay. The general structure of the antibody. You see here. So this is called a monomer. Right. And IgM is a pentamer. Apparently there are 5 monomers here. 1, 2, 3, 4, 5. Dia bergabung. So this is called a pentamer. Right? IgD is a monomer as well. Very similar with the IgG. Beza dia adalah tengok disulfide bridge ni. Untuk IgD, there is only one disulfide bridge. For IgG ada dua disulfide bridge between dia punya H chain. Nampak kan? Right? Itu beza dia. Right? IgA is a dimer. Okay, a dimer means ada dua monomer. Right? It can be in this horizontal or vertical arrangement. While IgE is a monomer. Okay, if you compare IgE with IgD and IgG, IgE has 1, 2, 3, 4 disulfide bridge between its H chain. Okay, tak? Sangat berbeza semua ni. So, IgG, alright, can be found in our blood plasma. Okay, 80% of this IgG found, okay, in our blood plasma. All right, they are roaming around, right, looking for any pathogens to be destroyed. Okay, to be destroyed or to be deactivated. Right, so this IgG is the most abundant antibody, right, in the primary and secondary immune response. We go to that later. And then this IgG can crosses placenta and provides passive immunization to the fetus. Right, so mothers, when they are pregnant, okay, they are providing their fetus with this IgG. Seperti saya cakap kamu sekarang pun lah, okay, under normal conditions, this IgG selalu berkeliaran dalam badan kita. Alright, IgG. Next, we have this IgM. IgM, okay, can be found on the surface of the B cell. Right, we go into that later on in the next video. Right, surface of B cell, as well as in our blood plasma. Okay, so this IgM, okay, since this IgM can be found on the surface of the B cell, right, this IgM is the first class or first type or first group of antibody, all right, that will be released by the B cell during primary immune response. Okay, maksudnya ada jangkitan saja dalam badan kita, ada patogen saja, first antibody yang di release oleh B cell ialah IgM. Okay, IgM, first class of antibody released by the B cell. Next, we have IgD. Okay, IgD. IgD can be found on the surface of B cell. Okay, similar with IgM. Now, this IgD, all right, it plays a role to activate the B cell. Okay, you will learn that later on, right, in the humoral immune response. And this IgD has low concentration in our blood plasma. All right, apparently this IgD only come into action, all right, when there is B cell activation needed. Okay, bila B cell itu perlu deactivate, baru ada B cell, baru ada apa ini, IgD. Alright, kalau tidaklah IgD ni. Kalau B cell ni tidak perlu diaktifkan, tidak IgD. Alright, when our body wants to activate the B cell, okay, IgD will be secreted. Okay, next we have IgA. IgA can be found in our saliva, our tears, okay, and then milk from lactating mother, okay, and other body secretions. Right, so you don't cry. When you cry, you are wasting your IgA. All right, so when we talk also, right, when our saliva is sprouting around, so you are wasting your IgA. So this IgA, okay, it protects our mucosal surfaces, right, and prevents attachment of pathogens to our epithelial cells. Okay, now for example, a virus, a flu virus, flu virus, when it enters our respiratory tract, what happens? This flu virus will bind and travel into or get inside our respiratory epithelial cell, right? So meaning to say our respiratory epithelial cell is already infected with a virus. So this is when the um, natural killer cell will kill directly our respiratory epithelial cells, because the respiratory epithelial cells is considered as already infected with that particular virus. Okay, so to prevent that thing from happening, this IgA, okay, will prevent any pathogens, right, to attach to our epithelial cells that eventually get into our epithelial cell, right? So this IgA strategically secreted into the respiratory, digestive, urinary, and reproductive tracts. Okay, who secretes IgA? Our B cells. Now IgE, okay, secreted by plasma cells. Remember, plasma cells are B cells I mentioned earlier. Okay, is IgE secreted by plasma cells in the skin, right, and tissues lining gastrointestinal and as well as respiratory tracts. And this IgE are found mostly in body fluids and skin. Okay, so this IgE, when bound to antigens, okay, this IgE will bind to the mast cell and basophil to trigger the release of histamine that contributes to inflammation and some allergic responses. Okay? So IgE plays a role in giving allergic responses as well as inflammation. Now, IgE has the lowest concentration in our blood plasma, right? And this IgE defends our body against invading worm or parasitic worms. But despite we have all these classes of antibody, our body still somehow managed to be penetrated or infected with pathogens as well as worms. Okay, so now the last part for today, antigen and antibody interactions, right? There are 5 antigen and antibody interactions here, right? So the first one is opsonization. Okay, I don't have the diagram here, right? So opsonization, right, is a condition where the antibodies bounds to the antigen on the bacteria and then present this antigen antibody complex to a readily recognized structure for both the macrophages and neutrophils for phagocytosis. So, like what I mentioned earlier, antibodies that usually bound to this antigen, right, to form the complex, right, usually the IgG. Okay, so IgG melekat di bakteria tu, antigen di bakteria tu, okay, untuk membentuk apa, antigen antibody complex, alright, so that this antigen antibody complex is easily recognized for the macrophage to be engulfed. Alright, so senang lah. So because this macrophage, they don't know is this the pathogen. They know this is a pathogen, but they somehow there are some pathogen that they don't really able to distinguish. So it's the job for the IgG to bind with that particular antigen on that pathogen to tell the macrophage, hey, you have to eat this. So the macrophage will engulf, right, that particular antigen antibody complex, right? Selain macrophage, neutrophil pun buat benda yang sama. Alright. Okay. Next interaction is the neutralization. Okay. So like what I mentioned earlier, in neutralization, what happens? Antibodies will bound on the surface of the pathogen. You can see here an example here. This is the virus, this is the bacteria. So this antibody will bound on the surface of this pathogen, right? Either on the pathogen or on the antigens. Okay, so this will neutralize the pathogen as well as toxin produced by the pathogen. Okay, and this will block the activity of the pathogens and antigens. And then these deactivated pathogens and antigens thereby promotes phagocytosis. Okay, now usually IgA, IgG, and IgM, okay, exhibits this neutralization interaction. Next, we have agglutination. So, antibodies have at least two binding sites. Betul kan? Ada two binding sites kan, huruf Y tu. Okay, antibody ni kan ada Y, dia huruf Y. So, there are two antigen binding sites here. 1, 2. Okay, so having two antigen binding sites, we allow the antibodies to cross-link antigens. Okay, to cross-link antigens by binding to the identical antigens on two different cells. Okay, nampak contoh yang ni kan. So one of the binding site bind to this particular pathogen, and then the other binding site bind to this particular antigen or pathogen. Right, the same goes for this antibody. One binding site bind with this antigen of this pathogen, and the other binding site here bind with the antigen of this particular pathogen. Okay, so antibodies cause antigen to cluster or agglutinate, thereby promoting phagocytosis. Okay, so meaning to say selepas pathogen ni sudah cluster, right, the macrophage will engulf this. Okay, this clustered antigen antibody complex. So IgM causes agglutination, right? IgM. Next, we have precipitation, right? So antibodies, okay, cross-linking with the soluble antigen, forming immobile precipitate of antigen antibody complex, all right, thereby promoting phagocytosis. And IgG responsible for precipitation interactions. All right. Now this precipitation only specific for soluble antigens. So opsonization, neutralization, agglutination, and precipitation. Okay, all these 1, 2, 3, 4 interaction promotes phagocytosis. Okay, promotes phagocytosis where the macrophage engulf the antigen antibody complex. All right, now the fifth interaction, complement system. Okay, now the binding of antibodies to the antigen on the surface of pathogen, you can see this is the surface of the pathogen, right? Okay, this is the antibody, binding of the antibody to the antigen on the surface of the pathogen activates a complementary system. Okay, the moment this antibody bind with this antigen, a complementary system is activated. Now this complement system will generate membrane attack complex. Okay, this is the complement molecule, right, or also known as membrane attack complex. This membrane attack complex or complement molecule, okay, will form a pore in the membrane of the pathogen. So let me rephrase. Bila ini antibody bind dengan antigen ini, complement molecule kena synthesize, dan complement molecule ini akan menyebabkan satu pore, alright, pore di permukaan membrane pathogen. Okay, so ions and water will rush into the pathogen. Ions and water will rush into the pathogen. Okay, causing the pathogen to swell and lyse. Okay, macam ni gambar aja ni. Pak, air dan ion masuk ke dalam pathogen tu. So ini akan jadi cell lysis. So whether activated as part of the innate or adaptive defenses, this cascade of complement protein activity results in the lysis of foreign cells and produces factor that promote inflammation or stimulate phagocytosis. Alright, so meaning say, okay, bila antibody ni bind dengan antigen ni, right, we have complement molecule, right? This complement molecule, as you have known, it will form a pore, right, in the membrane of the pathogen. Apart from forming the pore, right, this complement molecule also trigger inflammation as well as phagocytosis. So the antibodies that are involved in this interaction, all right, complement system, is the IgM, IgG, and IgA. Okay, now let's go on to these terminologies. So primary infection is an infection that develops in a healthy individual. That's primary infection, the first infection. Right, when we have first infection, so this particular first infection will develop. So that is the primary infection. Secondary infection means an infection that develops in an individual who is already infected with a different pathogen. Okay, so it's secondary infection. Now, immune responses to antigens, right, are categorized as primary and secondary response, right? This primary and secondary response, also known as primary immune response or secondary immune response. So primary immune response occurs when an antigen comes in contact to the immune system for the first time. So meaning say during primary infection, our body will give out primary immune response. So during this time, the immune system has to learn to recognize the antigen and how to make antibody against it and eventually produce memory lymphocytes. Now, secondary immune response occurs when the second time or third, fourth, fifth, etc. the individual is exposed to the same antigen. Okay, so at this point, immunological memory has been established and the immune system produces antibodies immediately. Contohnya macam chickenpox, right? When you have the chickenpox for the first time, you are having primary immune response. Okay, and then when you have the chickenpox for the second time or third time and fourth time, you have secondary immune response. And usually on the second in the second immune response, all right, for example of the chickenpox, you don't exhibit severe infections compared to the first primary infection, right? Okay, so now this is a graph that a graph concentration of antibody against time. Okay, concentration of antibody against time for the primary and secondary immune response. So in the primary immune response, all right, when the body is first exposed with that particular infection, all right, the concentration of the antibody is low, right, and then the interaction is slow, sluggish, and short term with long lag phase. Okay, with long lag phase. So during the primary response, okay, our body has more IgM than IgG. Okay, more IgM than IgG. Okay, so after secondary exposure, okay, means say our body is exposed to a particular pathogen for the second time, secondary immune response is activated, right? So the secondary immune response, they are quick, strong, and long term with short lag phase. Okay, we nampak ni lag phase dia sangat short. Okay, kawasan ni. Within this period of time, this high concentration of antibody is already generated. That's why short lag phase, right? And more antibodies are secreted. And in this secondary immune response, IgG is more than IgM. All right. If you compare with the primary immune response, they are slow and sluggish. All right, and short term with long lag phase. Tengok dia punya lag phase ni sangat lama. Okay, compare with the secondary immune response which has shorter lag phase. Now, it is advantageous for an individual to produce immunoglobulins of different classes, right, during an immune response because each class has different function. During a second exposure to the same antigen, while the memory cells are activated and secrete antibodies other than IgM, other naive B cells also recognize the antigen for the first time, become activated and secrete IgM. Okay. Memory cells can survive for several decades. All right. We go into that in the next video, right? But these memory cells, it can survive for several decades, which is why people rarely contract chickenpox a second time after they have had it once or have been vaccinated against it. So the vaccine triggers a primary response so that if the actual pathogen is encountered later, a large and rapid secondary response occurs and stops the infection before disease symptoms are even detected. Okay, so other than chickenpox, like the COVID vaccine, when we first receive our first jab, all right, this is the amount of antibody being secreted by our plasma cells. Okay, after 2 weeks, we receive the second injection, right? Second jab. So this is the amount of antibody will be secreted by our body, all right, to get ready for any COVID infection. Right, so it's very important for you to get vaccinated. Right, so that our body can produce this huge amount of antibody to get ready for any actual infection by the COVID-19 virus. All right, if you don't get vaccinated, what happens? You don't have this huge amount of antibody inside your body to combat with that COVID-19 virus. The moment the virus gets into your body, it will destroy your body already, your immune system, right? Before your immune system can even generate this high concentration of antibody. So if you get vaccinated first, your body able to generate this high concentration of antibody before that virus can even destroy your immune system. Okay, so that's it for today. Thank you. Bye.